Okay, so before I get started, I have to make an announcement, which is that Boris Schreiman, who was here last week, seemed to have liked this crowd very much, and he would desperately want many of you to come, if it is possible, to a school he and his friends are organizing in UCSB. So I got an email, and Mukund got an email saying, I should have done it myself, but there would be a microbial strategies and survival of evolution course running, a lab course that will run in parallel with a workshop on evolution of drug resistance. And the key things, well, here is his poster. Okay. Oh, okay. Please, yes. Okay. The key thing is that the application deadline is March 15, 2014. So you don't have, okay, you have enough time, okay? And it happens during summer, July 21, August 22nd. From my personal experience, I can tell you it's a wonderful place to be, both scientifically and every otherwise. So he was keen that, and you can see that there are several very esteemed scientists who are among the instructors. So anyway, so do keep this in mind. There will be flyers around. Apparently, some posters will be put in the NCBS campus. So I have done my due to him. Okay. So now back to our topic. Now, so I have to say something. These talks that I'm giving, okay, they are not a set of lectures I have prepared before and given to others. And I prefer that you think while I'm giving talks, as opposed to in many talks where you see slides after slides of results and data, right? 1.5 hours is too much for that. On the other hand, the risk of preparing things that way is that if you treat me with stone silence, okay, then, okay, I would immediately give you the answer, and then I'll get even more nervous, and I'll keep on going faster and faster. Then you will be very happy because it will be done in no time at all, okay? And something like that happened yesterday when I realized that I have a very, very, very busy slide, and then I'm sort of jumping up in there trying to write something, asking you something. So I apologize for that, and plus the Python snafu, okay? But I'll risk it again today. And please, when I ask you a question, do not worry about whether your answer would be silly, okay? Sometimes I'm looking for the silly answer, okay? And I'll thank you for that, okay? So, okay, let's get started. Okay, so you, so you have, you have seen much of that already. Sorry, I should have gone to the right page. Okay. So the key point that we tried to make yesterday is this, that, that I have this kind of, so if I add this penalty, I get basically soft thresholding, right? So if some variable without that penalty, you are estimating to be small, you will set it to 0, right? And the next key idea is hidden in the guts of this terrible slide, which is that when I have not one variable but many variables, these variables interact, okay? You get the same kind of soft thresholding, but what happens is that whatever the original x naught is, okay, it gets shifted by interactions with the errors in other variables, and then the rest is about how to estimate the size of this error, right? And I tried to argue that there is some fancy way of doing it, but okay, as long as you understand that much, we are happy, And okay, there is some little chest thumb that this works. Actually, much of this setup, it's not our work. It's been looked at, the sort of replica analysis was started by a Japanese group a few years ago. And it's been taken up, and everybody, at least in a certain community, uses it. But key point I was trying to emphasize is the importance of other confusing, right, correlates, right? When we have, as I said, I have multiple sites, and I have many, many, many such sites. Quite often there is accidental overlap of certain directions with the data, and we'll actually see that come through a little bit better today, okay? So here, okay. So few things to clarify before I go ahead. So I think today, yesterday I got a bit flustered suddenly realizing that, oh my goodness, maybe I don't have enough time. Sorry, I actually have too much time, okay? Today I shouldn't worry. I'm going to see how far we can go, what we can discuss, and see whether I can engage you. Okay, so few informational things, okay? I keep on using the word lasso for this L1 penalized regression. But in mid-90s, when lasso was invented, okay, by Tipshirani, it was not meant for solving underdetermined problem. It was meant as a garden variety regression method where the small terms are not really noise fits, okay? So you want to throw away things that are really dominated by noise, right? So sometimes, and the whole adaptation we have for this, okay, sorry. Somehow it has decided to freeze on me. Okay, forget. Okay, anyway, so this quadratic things plus whatever we've done, there is one extreme example, remember, where I'm just minimizing the norm subject to y equal to hx. Okay, that's often called basis pursuit. Okay, and the quadratic analog of that is called basis pursuit denoising. Although mathematically, the function you're optimizing is the same. Some people call it by another name. So you should know that essentially it is the same formulation. It's just that in one case, the emphasis is on underdetermined systems. And in the other case, it's sort of garden variety regression, okay? But it's effectively the same thing, right? So that's one thing to keep in mind when you see many papers. But I think I'll go back to something we started out long time back when we were talking about for example, QTL analysis, right? And what I told you is that in QTL analysis, right, you have this, I dare not go back just in case this thing freezes. You go across the genome, you see snip, 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 snip. You do each case a test. You say, what's the log odds that this thing explains well some phenotype, okay? And you get a graph. There are a little bump here, nothing, or noise, bump here, et cetera, et cetera. Now, you can tell me that I can take this kind of an approach. Let's do this test. Let's take like the top 10, okay? And why do we need this all complicated L1 regression and whatnot, okay? So here is think time 1, okay? Do you think this is all this lasso or basis pursuit type of method, which seems algorithmically complex, right? Not very complex, but still, it's much easier to do one thing at a time and figure out the few peaks, right? That's how most things are done in life. Why not do that? Okay. So any ideas? Okay, you are going to treat me with the same degree of disregard, clearly. Okay, so let me do something. Let me get out of this stupid thing, and let me see whether I can show you something where hopefully, so this is our same little IPython window. So last time, what we had done, we have created some X0, okay? Here is the x naught, very, very sparse, okay, right? We had, if I do it again, I'll make enormous number of mistakes and we'll suffer, right? But you remember there is sort of this lasso-like algorithm solving for x. This is very sparse, okay? We have some hope. It is some, I guess, a 200-dimensional matrix with 5 non-zero things, right? So I have actually gone through and created something, and in this, as you saw last time, in this C of 10 to the minus 6s and 10 to the minus 5s, that's just because only so many iterations have been run, there are some 10 to the minus 1s like this, like last time, okay? And now, okay, we know what to do. We usually do this. Okay. And we declare victory, okay? We say, okay, look, again, I'm still very lazy to add things, axes there, but this is all zeros falling on zeros, right? You see that the estimations are not perfect. A perfect estimation should be on this straight line, diagonal line, right? But more or less, 5 things were non-zero, you picked up the 5 things, okay? Now, I could say, you fool, you have been working too hard on it, okay? Let's do something else, okay? Find 5 variables, okay, which have the best correlation with the data. Suppose I want to do a 1-variable model. What's the best model? What's the second best model? What's the third best model? What's the fourth best model, right? Okay? So think again. What I have is a y vector, right? And each x picks up one column of my matrix H, and the regression result, okay, if it's one vector at a time, would be just a particular column of the H matrix times x, right? So a line, right? So you can just look at which ones have the best angle, right? The closest angle with the data vector. So I have data vector, I have various single variable fits, right? So the most significant ones are the ones with the least amount of angle, right? So I can look at dot product, okay, and try to organize the dot product. And I can see whose dot products are the biggest, right? Absolute value-wise, either positive or negative, and pick those. And then see what happens, okay? So we try this, okay? Hopefully, again, I don't run into something. So this one, I actually calculated the dot products just because I have no faith in myself anymore. And hopefully, the dot products are called Z, okay? And I suddenly have this. Okay, so what's what here, okay? The x-axis still is old x0 values, okay? When these points all have x0 equal to 0. Remember, there are 5 guys which have non-zero values, okay? And these are the cos theta of the angle, okay, between a particular variable, okay, and the data vector. Basically, the corresponding H matrix column and the data vector. Okay, if I pick the strongest 5 correlations here, who would I pick? I should pick that guy. I know that's a good guy. And 5 is very sparse, remember, okay? I'll pick that guy. That's also a good guy. I might pick these 2. Okay, so in this case, I've almost done right, but you will begin to see problems coming your way. Next, I should pick this guy, some dot point here. And what has just happened, right? I actually won't pick this guy because there are many, many zero fellows, okay, whose vector has better angle, okay, with my data point, okay? So I'll miss this, okay? I'll actually pick that. You will tell me, okay, 1 out of 4, okay? 1 out of 5, okay, not bad, right? Well, actually, no. Tell me, okay, if I do now the following. I keep the same vector. I pad it with more zeros. So instead of 200, now I'm 250. I have 50 more zeros added. Okay? And I take my H matrix and I pad a few more random columns. Okay? So I now have a bigger problem. I have 5 observations and 250 size matrix. Okay? And I do dot products. Okay? This is where you have to turn on your mind's eye and think. Okay? What would happen to this picture? This time I'm going to ask you questions. And you guys are going to sit even further and further behind, right? But you don't have an option. Okay. Yeah? More dots along the zero line, right? Because that's the only game in town. As more dots happen along zero line, the overall width of these confusion creators typically increase or decrease. If you have more, well, if you're lucky, it falls there and doesn't do any more damage, right? But otherwise, it keeps on growing. That is already what has happened, okay? The 200 minus 50, actually, okay, so where is all this coming from? Okay, remember, if I take, okay, R direction, and I take another random vector, the distribution, right, is some well-known distribution. That's sort of that overlap, right, is very peaked around 0. We already said that the size of typical overlaps is order 1 over root m, right? So if I said that this is a 1 over root m standard deviation Gaussian, we are fine, right? Okay? But as I sample more and more and more and more from there, the extreme values keep on growing, right? So if even from a very narrow distribution, you have sampled many points, okay? The biggest on positive and negative side keep on spreading, right? Now, if I have the following situation, okay, now if I really had k equals 1, right? I had one variable, I produced my y with that. Of course, you cannot beat that, right? Because in that case, that very column has really, they are perfectly aligned with the data. The cos theta of these two vectors would be 1, and nobody can beat 1, right? But, sorry.
Suppose you find the very, okay, not even one variable case, but suppose you took the biggest one in this.
Right.
What would be the coefficient that would apply? So it's not exactly cos theta is not 1.
No, I'm actually even going to do the following. I'm going to order. I give you a list of the top one to the next one to the next one.
Find the top one, you subtract its effect, and then do the next one.
I'm coming to that. That's my next point. Okay. So here is just one at a time, and realize the LOD score looking is one at a time. Okay? That is practice. You get big PhDs out of Harvard doing population genetics that way. Okay? So, right? So all the gurus of the world live off that, right? So here I have, So if I have a single guy, of course, if I had one guy, that guy would be 1, and spread as is may, it will not catch up. But if I have k guys, if I took y to be a combination of k things, right? Okay? What's the size of the vector? So say each x is order 1. Okay? I take k vector linear combinations with random vectors. So I've been asking you to do a lot of things in your head, which is maybe not fair, right? If you ask what the square average of that, you will see that this, okay, maybe I should try to do this on the blackboard in this very blackboard unfriendly environment. Okay? So I have x1, h1. Remember, these are the columns, all the way to xk, hk. Well, on the average, of course, if I go over all possible h's, the average is 0. But I would like to know what the variance is of this object, or the size of this object, right? So I should just do a square, right? And it's pretty much the same math 100 times over again. I end up getting x a square, okay, h a square, sum over a, and the cross terms. okay? And I'm asking, say even I fix x, if I took average over hs over random matrices, right? What would this be? You are too sleepy in the morning. Okay, I scale each component of h to be order 1 over root m, right? So square of each component is order 1 over m, and on the average, therefore, sum over m components is order 1. That's my scaling. So h1 square, h2 square, h3 square are all order 1, right? They are almost like an orthonormal basis. What about h1 dot h2 averaged? I pick 2 random uncorrelated vectors. What is their dot product average? Who said? Okay, 0? Okay. Huh? So, okay, that doesn't exist. So how many terms? So of course, A here is only running from 1 to k. Okay? So this vector, this is actually y, right? Its size is of the order k term summation. Of course, I'm assuming each x is order 1. Each non-zero x is order 1, right? So the norm of this vector is order k, right? What is the size of each component then typically? If I, sorry, stupid me. Now I take this vector y and scale it down to be norm 1 so that it's a direction. What would each component be then? It will be 1 over root k, right? So as k increases, if I normalize that vector, the components become much smaller, right? Right? So typical overlaps, typical coefficients in the normalized version would be order 1 over root k, right? If k was 1, I'm safe, right? And then as I sample more and more things from this Gaussian here around 0, okay, the maxima among them would very soon be into the edge, okay? So that typical size of entries there is going to be smaller than the biggest ones, okay, that came completely out of random, okay, overlaps, okay? So this is the reason why, unless you are extremely sparse, right, the method of 1 at a time, okay, soon runs into trouble. Okay, so I told you this runs into trouble. Does the thing? Let me go back to this. So what Mukund was saying is that, well, it's a bit foolish to do so 1 variable at a time versus this kind of L1 non-penalized thing. L1 non-penalized things wins every day, okay, unless you are very, very sparse. Okay, because of this thing that if I have a big matrix, lots of zeros, some of those vectors are actually by accident very well aligned with the data, and sometimes, okay, your original if it's a multivariable effect, right, the individual effect of a single variable is actually smaller in absolute terms than a random overlap. Okay, so the next thing to so that's what I actually said so far. Okay, next thing is to do stepwise regression, right, which is the following. Okay, I take the one variable that explains most of y, the guy that's best aligned with y. I find its best, okay, point, and again, these are a little bit better with pictures, and I should have made the pictures. So in some arbitrary place, this is the y vector, okay, and one of these guys, call it some h vector, or why am I writing vector for this and not for the other, okay? Or maybe I put vector for both, okay, right? You have basically, right, you find the point, okay, that comes closest to it, right? And then you say, aha, this is my best direction, it's included in the box, okay? Now, let me try to figure out what remains, okay? So this is my residual vector. You now say, of the rest of the vectors, who will be my guest in trying to explain this? Okay? So maybe some other poor fellow shows up its hand, it's pretty well aligned with it. You kind of go up there, you find your next approximation. Okay? And usually, what you do is that you run some kind of t-test to say, if I had absolutely random data, y was generated kind of out of noise or some signal plus noise, if I am in the noise territory, okay, then by adding this, have I done a significant reduction? Okay? So you do some T or F test, okay, to determine whether you are really improving. Okay? At the risk of getting another sort of very silent moment, okay, what might be some of the problems with it? Okay? Suppose you are writing a code to do that. Actually, this is done in practice for many problems. What would be the few things you have to decide? Just think you're sort of cooking up an algorithm. For example, if the first variable you choose, okay, you will do some test to say which is the best variable, right? That's kind of easy. And whether it's significant, even should I include even one variable, okay? Then you try the remaining variables, a set of tests, right? What would be some of the things we have to think about when you set up a test? Okay, I'll give you a hint. When you set up a test, what is one thing you have to decide? Say a t-test, okay, or an f-test. A threshold, right? How do you set the threshold? Anyone? 1% is good? For example, we usually do 1%, 1,000. Well, don't nod your head. If I ask you that question, it's most probably there's a trick there. Okay? So, how do you decide? What is the threshold you will choose? Okay, how much is noise? Okay, so let me actually give you even a concrete thing, and then maybe it helps you. Okay, I have 200 variables, right? I had, I don't know, in that example, maybe 50 data points, right? I have to choose which of these 200 variables should I include, right? And is it significant enough? Okay, is a 1% test, T or F test, good enough? Okay, what does the p-value of 1% mean? That means, had there been nothing, okay, there is no signal in the data, a random test would say yes, okay, 1 out of 100 times. Okay? You have 200 variables. Okay? How many times you are supposed to get yes? Twice on the average. Okay? So this is the problem of multitasking. Okay? You try to do it once, right? If you have many, many tests running parallelly, right, you actually need stiffer thresholds. Okay? Now, I have not only a multitasking issue here, I have a nested multitasking issue. As I go down, okay, then I have to, dependent upon the first test, what it said, you have to do something else second time. And so it gets a bit messy. Okay? Now, people have tried to come up with some ideas of some complexity penalty, this, that, right? Try to correct for multitasking. Turns out that the very strict complexity corrected multitasking at low sparsity does a good job, but does much worse than L1. Okay? Now, this is maybe too many things I'm saying by hand waving and not telling you anything. So look at the trouble, though. In many problems in biology, we have to run thousands of tests. In this case, 100, but in many gene experiments, you run thousands of tests. Okay? One way to say is that you like 0.1 or 0.01 p-value divided by 1,000. That's kind of the Bonferroni correction, right? But then sometimes it is too stringent. It's saying I would not even make a single mistake. Okay? Or the probability of making a single mistake is really, 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 okay, lower than 1. Okay? So you feel very confident. Maybe you in the lab or in your biologist avatar say, I don't care, we often make wrong mistakes, right? Or many, many mistakes. So maybe if 20% of your results are wrong or 10% of your results are wrong, I don't care. Okay? So that's a different approach, a little bit kinder approach, okay? That goes by the name of false discovery rate. So you say, my tolerance is that I can take 20% of garbage, okay, or 30% of garbage. I don't want everything to be garbage, right? So I allow some of the tests to go through. If you do that with this kind of stepwise regression, okay, you kind of do okay, right? But then the okay is also qualified. What happens is that it also has a threshold where it starts failing, just like previously L1 norm had kind of this threshold where it fails. And they are comparable, but when it fails much more spectacularly. What do I mean? I showed you this kind of error as a function of sparsity or change of measurements, and you see a continuous rise. There you see a much sharp jump rise, okay? So long and short of this is that stepwise regression works depending upon how you set up your thresholding scheme, okay? If you do it with the right way of doing FDR, and false discovery rate. But then when it doesn't work, it produces much bigger error. Whereas in some sense, when lasso doesn't work, close to the threshold, it produces kind of gentle mistakes. Yeah.
But the thresholding is one issue.
Right.
See whether that variable should be included.
Right.
It looks like once you found the variable that might be important, the size of the coefficient, to estimate that correctly would make a big difference to what the direction of the other one.
Okay, so you're very perceptive. Okay, so the trouble of the greedy approach, okay, sorry, of my capitalization, is exactly you end up committing to the size of how big you are, and then you come in. Okay, and this is the source of some of its troubles. Okay, now let me actually, okay. So I was just going to say something about ridge regression. Okay, so the short answer is that suppose the way to go around that is to do it incrementally. And then there are two methods, okay? One is called LARS, okay, the least whatever angle regression. And there is something called stage-wise regression that exactly does that. It increases a little bit in the right direction, then asks the question again. And increases in the right direction, and the answer of, I just don't want to be talking to you, but the thing is that those two methods and lasso pretty much morally give you similar answers. Okay? So lasso is in some way more modulated, not doing this jump, 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 but actually do it along a curve. Okay? Right? Okay. We had some discussion about another way of doing this penalty. And that's what's called the most traditional shrinkage method. Shrinkage is basically, I got some excess, I want it smaller. Because maybe if there is something there, I'll have a little bit of bias, but generally it's being dominated by noise. Shrinking makes the variance less. So you get a little bit bias, but you actually reduce the variance. Okay? And that should be lovely after all this complex optimization, because this is just a quadratic problem. We know the answer to that. You can write down explicitly what the solution would be, H transpose Y, this kind of some in matrix. Now, H dagger H, remember, okay, it's a rectangular matrix. You do H dagger H, you end up getting a very large matrix with many zero eigenvalues. This is why that is another way of saying that you cannot do the naive regression formula, which is H transpose here and just H dagger H inverse. You cannot invert the matrix because you shouldn't be able to. There is indeterminacy, right? There are some directions you can put anything and you get it. Having this regularization term introduces a little C times identity there, and suddenly everything is good, right? And you get an answer, right? Now, what's the trouble with that answer in the context of sparse regression? That's the following. Okay. I think I already said what it is. Any of you remember, or any of you have a thought why this may not give you sparse answers? Okay. Okay, so there is actually a way of thinking about rectangular matrices. Many of you have done that in principal component analysis or something. It's singular value decomposition. So, and I really apologize trying to write this matrix with Microsoft equation editor. Half of I think my life was shortened by several years because of that. You end up writing the matrix H, the rectangular matrix, as some unitary matrix, M by M, then some rectangular matrix of the same size, and then another unitary matrix here. Okay? Now, what's going to happen here is that there is going to be only M non-zero diagonal entries there. Everything else would be zero, right? So what this, and these are kind of daggers or transposes, okay, if there is complex numbers, we take complex conjugates, of a bunch of vectors in this n vectors in this n-dimensional space. Okay? And these are a bunch of vectors in the m-dimensional space. They are all orthogonal to each other. Okay? So what this basis does, okay, is that if I write my x and y in that basis, right? In that basis, action of H is very simple. If you are one of the first m components, depending on which component you get multiplied by Si, right? So the ith component gets multiplied by Si. And anything beyond them, you just get squashed. You become 0. So what has happened, because n-dimensional space is much larger, there are directions that are basically the kernel, right? The thing that would become 0 when you apply with the operator. And there is a space where you get a non-trivial answer. You have an orthonormal basis that splits, right? The first m, okay, are something that produces a non-zero answer, and rest are the orthogonal directions to that. Okay? So in that basis, if you ask for what the answer is just this. Okay? For the first m, the component is going to be some SB times W with some psi-dependent answer, and rest of them would be 0. Okay? Now, that means that when I do this singular value decomposition, I get this big n cross n matrix, of which I can read off a bunch of vectors, m of which are the directions which have non-trivial results produced, and rest are just orthogonal to it. I'm not going to produce by ridge regression anything orthogonal to it. All the answers are going to be linear combination of these first m vectors. And the point is that if H is a random matrix, these first m vectors are practically a randomly chosen orthonormal basis. Okay? Right? So as a result, their linear combinations typically are not sparse at all. Okay? So I have done it, but I haven't gotten sparseness out of it. Okay? Fine. So okay, maybe all is not lost. There is something good about it. So here is my summary, and I'll leave this lasso type of thing right behind with it. The first two was I already mentioned in response to Mukund's question, and had to do with this issue that the greedy approach, when it added a direction, a particular variable, it immediately decided how big the variable would be. But if you do it in steps, okay, with some slightly different criteria, you get this first two method, stage-wise regression and LARS, and they are not that different from lasso in most practical problems. Okay? There is a method that tries to marry the good things of ridge regression, which is that it is somewhat stable to noise, right? It is somewhat robust. And lasso, which is that lasso ends up picking a sparse model. And if you choose the two penalty term coefficients properly, you can sometimes get the best of both worlds. Okay? In cases where lasso is sort of mistakenly choosing a particular variable to represent all the influences, when there is another variable which can about as well explain anything, it chooses actually a linear combination of the two. This is something also methods like LARS do. Because I have two vectors, and I have the data vector. Rather than choosing one of them, you quite often have the least angle guy in the middle of the two. Okay? So there is something you can get out of that. And then there are a whole bunch of very different methods, okay, called belief propagation. I'm not even going to try to explain what those are. These methods give up on the idea that I will try to solve some well-defined, okay, convex optimization problem. Okay? Those methods are basically sort of inspired by the idea that, okay, if I try to solve, remember I said if I try to solve the original problem, okay, I'll be dealing with some function that is non-convex. Okay? I could try to sample, right, with this probability distribution idea from an energy that is non-convex. Now, usually what happens is that if I do the naive Monte Carlo there, okay, then it often takes long time to converge to something. On the other hand, what I tried to do, using the fact that there are a lot of variables, et cetera, I tried to write down the equivalent of what will be mean field equations. I tried to write how does average x here get affected by average x there, and there is a better chance of trying to solve those equations directly, okay, by some iterative scheme. Nobody guarantees that they would for sure converge or something, but they have better hope than just doing Monte Carlo. Okay? So these are methods that try by various crazy iterative things to get something like the mean field solutions right away. Okay? And they actually, in practice, succeed in reconstructing the vectors over a region greater than lasso, typically. Okay? But it also becomes very, very dependent on system details. Okay? But, okay, so there is a whole world of bleeding edge, okay, activity in that world. Okay. What is it? Huh. Mm-hmm. Right.
But you can also start to eliminate the variables that you think will not contribute to the system. So if you can actually start to fix them, does the problem, the chances of identifying the ones that contribute improves?
So you are saying start from another end. Can you say who for sure doesn't contribute? And I'm going to say that that is not as obvious as one thinks, right? So, yeah, let me see. Okay, why? I think the issue is this. Okay, if you look at one particular vector, okay, and you say what's your, so unless it's kind of totally orthogonal to y or something, right? Then you are in luck, right? You say I'll eliminate it because you are kind of small, right? But it could be that actually the significant answer is that several small effects together among many vectors. It's the same thing we talked before, right? Remember there was this picture where there are all these zeros which had pretty good overlap, and then some non-zero ones actually has smaller overlap than the extremes, right? If I increase k, they will get closer and closer to zero, right? So if you start eliminating a bulk based on how small their overlaps are, so maybe what you have in mind is something more sophisticated than that. You have to look at their group properties, right? And what makes it difficult is that you have to choose sort of an active set, and there are too many active sets. So hold that thought. I don't want to say there is nothing in that direction. I haven't thought much of it. I don't see a naive way immediately of doing it. Okay? Okay. So, yeah. Mm-hmm. Right. Yes. Right.
Typically, what I heard is that you use L1 to do model selection, and then you remove L1 to estimate the coefficients.
Right. So, yeah, right. So if I had no noise, which is, I agree, is rather irrelevant for many problems, except in some group testing problems where you can think of this as no noise problems. Okay? Many problems with noise, I believe that's right. We are better off doing some variable selection and then doing a better noise resistant regression.
What you should really solve is L0 problem, which
Right.
is non-convex.
Right.
You say, well, I'm not going to solve that. I'm going to solve the L1 problem, which
Right.
is this convex.
Right.
And this is why I believe correlation methods
Mm-hmm.
get solutions where
Right.
L1 does not, right?
Right.
So, but then one way of eliminating this distortion on the estimate is
Mm-hmm.
reducing whatever the subtotal is to.
Okay. So.
Who use L1 to do model selection and then.
So it depends on which regime you are in. Okay? Suppose you are in the other side of the phase transition for L1, you don't need to work that hard. Okay? You actually solve the basis pursuit problem, which is lambda going to zero limit. So as lambda goes to zero, and if it was really noise free and you are in the good construction regime, there is no distortion. Right? So if you solve the LP problem in that regime, subject to the constraint, you should get the full answer, correct answer. Okay? That's the claim, right? Now, what you may be right is that if you have gone past the threshold, could you do anything better? But past the threshold, remember, I actually have some mistakes in choosing my active set. So you need a more inclusive active set to be able to do things right. And I think belief propagation algorithms, the good thing is that it's not sort of completely giving up on them. Okay? But it's a good question, right? Whether there is some way of starting with So, for example, the FDR version of even stepwise regression, okay, can sometimes go past the threshold of L1 and do it. Okay, but then a little bit later it produces horrible mistakes, right? The transition almost looks first order there. Okay, but so I haven't fully answered your question whether there is any way, because I haven't thought too much about it. I can see in some limits it's not an issue, right? I always kept lambda finite just because I was running it in front of you. Okay, but if I really was clever enough to hook up a good LP solver with it, okay, I would not in good regime get those distortions. Okay, and it's actually funny. NumPy or SciPy doesn't have a nice native LP solver, so I looked for one, but I decided not to take chances. Okay. Okay, so I change gear. Okay, here. I start talking about something very different, and hopefully, at the end, come back and the two subjects meet. Okay, so well, again, there is lots of data. The problems I told you about, okay, is that I have some output, like it could be binding of some protein or some kind of gene expression phenotype or some disease phenotype, right? And you are trying to express it in terms of many possible variables, okay? And there are too many of these variables, but hopefully many of them are zero, right? And we say, I hope it is sparse, and I find some answer, right? There are many other cases. You actually are given a lot of data, and someone is asking what kind of probability distribution this data came from, right? Now, if it is a very high dimensional data set, we'll never get enough data to populate the space enough to have kind of multi-dimensional histograms, right? Or where some little element by element I'll get that, right? So most of the time in the data exploration state, you are trying to guess some kind of probability distribution, make some hypothesis, move somewhere, do something, right? Now, the problem in the beginning has the same superficial look as a sparse problem, as the underdetermined problem, rather. And this is what is some issues related to so-called maximum entropy method, which is what I want to start with. So in many cases, I want probability distribution of in some high dimensional space, or it doesn't have to be high dimensional, for some x. What's been given to you is some kind of average measurements. So I have some quantities fi of x, some functions of this variable x. You give me these average values, right? And if you ask me whether you can solve it, typically the answer is no, right? You can actually think of this pretty much in the way we are talking about it. These measurement averages, if I give you precise averages, are really px times fix summed over x, quite often integrated over x, but let's think of them as a glorious sum. And if I have that arrangement, you can think almost x as an index, this being a vector, fix being a matrix, and I have very few observations to determine a very high dimensional thing, right? Now, given what I've been doing right now, you might say, aha, now I'm going to find a sparse solution, okay? But that is kind of counterproductive. So imagine I give you the average of the x vector, okay? I ask you for a very sparse solution. What would you choose? Huh? The most sparse thing is to take the average, put all the probability there, okay? Take a one-dimensional thing. I give you the mean and the variance, right? And I ask you to choose a very sparse distribution, okay? What would you do? Hey, no. There's something sparser than that. Think of them as if you have some grids and a vectors. What would be very sparse? How can I construct the mean and a variance and with as few non-zero probabilities as possible? Somebody showing two things, right? I'll take that as an answer. So I have two peaks, okay? Pretty much maybe equal weight. I have a mean in the middle, and I adjust the distance to the variance, right? Now, those very spiky probability distribution is exactly what you don't want to do, given some data, okay? So I now need the anti-sparse, I want a nice, smooth, fat probability distribution. In fact, maybe the fattest probability distribution. Okay? I want to ask, if you give me this constraint, how can I get I would like to get a bound on things, right? Now, that's not possible, although in some positive variables, if you know the mean, you know certain things, okay? And the variance, you know certain things about probabilities and tails, right? So we need to figure out sort of how much variability can I allow given certain means, right? And one way to think about variability in a probability distribution is think about entropy, right? So there is sort of this whole almost church, right, where the kind of the key figure, the key prophet is E.T. Jaynes, okay, who wrote a bunch of papers in mid-50s, which he sort of had this idea about statistics and entropy and physics and everything being somewhat similar, and some of this is almost theological, and others are somewhat useful data exploration things, depends on who you are. There are people who would say E.T. Jaynes is more profound than all physicists together in last century, and others would say, okay, he's a nice guy, he taught us something, okay? But what is the kind of problem he would say you do? He said, okay, put some Lagrange multipliers, forcing, okay, averages of this quantity to be m. There should have been an I index there, which I've forgotten. But subject to those constraints, okay, get the most, the highest entropy distribution, right? So what's the trouble of, for average mean, our spike answer? That guy really has how much entropy? If I all probably, it's okay. It's continuous, it's a little bit tricky, but if it was discrete, right, you will get 0 entropy because all probability. So in that case, Px, there will be only one x, and Px would be 1. This Px would be 1 inverse of 1 log 0, right? If I had 2 spikes explaining everything, okay, how much entropy is that? Okay, that's basically log 2, right? It's number of things possible equal to, right? So, right, so one of you said Gaussian. Gaussian is doing better than that, okay? It has a, okay, we have to check, but hopefully it's doing better than that with the right variance and all. And so let's see what kind of answers you're supposed to get. Well, okay, many of you know this already, so maybe it is not worth doing it, but the point is that if I try to vary P of x and try to find what the condition is, varying here, okay, produces 2 terms, but one is constant. You get basically log P inverse x, and varying here produces Fix, and somewhat magically, okay, once you solve it, Px becomes e to the minus these Lagrange multipliers times Fix, right? So you actually get something that is, as you can see at least, okay, physicists seem to like it because it looks like some kind of generalized Boltzmann distribution. I chose beta I to be the name of the coefficient so that it looks even more like physics for those who are in it, but people who are not in it, it doesn't mean anything to them. So lambda and beta are the same thing for you, okay? But these guys are, of course, knobs that you will now can play with, and if you play with it right, you will be able to match those conditions that the average is going to be the same as what is the observed mean, right? So, for example, if I had a variable that is non-negative, only lives on the left half, okay, right half of the real line, right? And I tell you the average, the maximum entropy probability distribution would be, okay, would be e to the minus whatever function I've averaged, which is x, right? So it will be like e to the minus lambda x. It will be exponential, okay? It's one-sided, right? And then I choose that coefficient carefully so that the mean is exactly what you told me it is, right? If you tell me, so, okay, again, don't freeze on me. Sorry. Yeah, so the betas are adjusted to do that. Okay, this is an exponential distribution. If I have mean and variance given, you can guess it will be a normal distribution, right, whose mean and variance would now be adjusted to whatever the values were. This is where some people get excited. They start looking at higher dimensional things. So for example, there are many cases you can have a discrete distribution of many variables. Okay? One favorite one for physicists is things like spin or binary variables, either 0, 1 or +1, -1, right? And suppose I tell you about average values of those variables and their correlations. Okay? So in that case, okay, the probability distribution that you get by reconstructing the maximum entropy one looks very much like a magnetic model called Ising model, where the spins are interacting with some couplings. This is some interaction between I and J. And there are these fields which are kind of trying to make SS go either towards the +1 value or the -1 value. So this kind of probability distribution, people have a lot of love for and intuition in certain community, and they would like to sort of say, oh, now I can tell you about other moments I can do testable hypothesis or something. Okay? And there is already quite a few papers, I just picked 2 random ones, right, trying to apply this method, trying to say maybe this will discover important interactions, biomolecular interactions or physical interactions. For example, there is a lot of data where in neuroscience where someone takes a multi-electrode system, sticks into either a mouse brain or somebody, or even a human brain sometimes, okay? And you get a whole bunch of numbers come up. So your big train of spike here, spike here, spike here, spike here, right? And some are correlated, right? Usually, correlations could be because they are actually talking to each other, or it could be because A talk to B, B also talk to C, and A and C are not really directly talking to each other, but there is some indirect influence, right? And this kind of model, right, the kind I described, perhaps is trying to disentangle that. So you can say, maybe I learned something about who is correlated with who, okay, who is really interacting with who, as opposed to who is correlated with who, okay? And this was another piece of work where people looked at diversity of antibodies and then look at sequence correlation there, okay? So, and you are hoping that they actually have some kind of biological significance, right? That these amino acids and those amino acids together mean something, okay? They have some kind of function, right? So there is, what? Anybody ask a question? No. Okay, so what is the trouble then? Well, the examples I started out with, right, which I said, oh, so one variable there, I give you the mean, and I know it's one-sided. You say, try the exponential distribution, okay? First of all, I can do a few experiments and come back and tell you whether exponential distribution was a good idea at all or not, okay? But if I had quite a bit of data, I know the mean pretty well, okay? So I can say, oh, you got trustworthy estimate of mean. At least that is not in doubt, okay? I got a mean and a variance out of a large strain of data, okay, maybe then even, okay? I have reliable moments. So usually I have often few parameters which are well estimated, and I actually then go out and try to construct the probability distribution, and then I can do anything I like. I can try to test whether that's a good probability distribution. I can try to make other predictions, see whether I'm right or not, etc., right? But now I have pushed this on to the world of kind of biological big data, okay? Which means I am, like in the previous example, okay, here, I am really asking for about n² correlations to be measured reliably, okay? And then I'm fitting n² parameters to it, okay? So in many problems, you are actually fitting about 100², okay? In kind of problems that are already under control, sort of people say that, okay, they can get somewhere, right? So I'm actually fitting bigger and bigger number of parameters. Again, I'm back to this issue that the generic model have many possible interactions, okay? My data cannot, at least for some of this interaction, I'm sure if I make up this model, I most probably would end up fitting noise. Okay? So is this a good idea, really, to try as many moments there are that could be gotten out of it, that many parameters, and I go fitting? Okay? And what should I do, right? in that regard, okay? So I could tell you a little bit. I don't think I have a full answer to it. I can tell you now a story in which we ran into this problem, okay? We said in the beginning when we went into the problem, we thought that, oh, there are all these greedy, get one variable first, get the next important variable first way of doing things, and those should suffice, and we suffered horribly, okay? And then you really, really want to go back to something that's vaguely principled, okay, and gives you a systematic thing. So I'll just briefly tell you about this problem, and you have lived here without biology for a very long time. So this is time to sort of introduce you about some biological problem that actually we have been working for some time, and hopefully get you somewhere, okay? So this problem has to do with chromatin and chromatin conformation, okay? And this is some obligatory introduction to chromatin. There is DNA, there is histones, right? This is called 10 nanometer fiber, 30 nanometer fiber, and this is sort of artist fiction, okay? Those molecular biology books need pretty pictures, okay? They paid somebody to make up these things. You know, they somewhere in very short letters would say, this is what we imagine it to be, but no, there's no proof, right? So for a long time, that was the situation. We didn't know anything. And this is very iconic representation of a chromosome, right? But if you know a little bit of biology, you know that iconic representation holds only for a brief while of the cell's life, okay? Just before, you know, they are to be separated into two different cells. If you put DAPI on the nuclei and look under the microscope, usually you see a blue fuzz with some little punctate things here and there maybe, but that's it. So this is actually someone showing in an onion cell, I guess, that this is things everywhere and these are separated chromosomes, and that happens kind of here. But most of the time, things are spread, okay? All DNA is spike, but what I want to focus on is that what is functional chromatin looping doing, okay? And the reason for it is the following, is that, you know, in many bacterial examples, what you know is that if there are regulatory proteins bound, you know, you can just look around there and find the gene it's most probably trying to affect, right? So it's within few hundred bases typically, right? Whereas in higher eukaryotes, you'll often find a regulatory site there that has to interact with something like a megabase away, right? And there are often other things, these pictures actually that of an insulator, they have to loop with each other to stop its influence from spreading. Maybe there has to be some other things, right? So there is very complex chromatin folding, as some people say, right, which has biological significance. And it's important to sort of figure that out, to see when a certain gene is being regulated one way versus another way, right? So there is some techniques to do that. And there are various 3 Cs and 4 Cs and 5 Cs and God knows how many Cs, right? But the broad idea is the following. Chromatin does some kind of looping. Somewhere things touch. You kind of get it stuck by some chemical trick, okay? Then you cut the chromosome up, so you get this kind of joined pieces, right? Looped pieces. And then you take these stuck pieces, somehow ligate them, do sequencing, PCR, whatever you want to do, depending upon the method. And you have some information that will come out if it is for a particular region, would come out in this way, as a big matrix, okay, of numbers telling you whether a certain location of chromosome is in contact with some other location of chromosome. You will get some quantitated number. It could be number of reads that were sequenced where one leg fell here and one leg fell there, right? It could be some smudge from some whatever PCR, okay? So you have a bunch of numbers telling you something about how strong is the interaction between site A and site B, right? So now let us take a step back and think what we are going to do there, okay? If it is something like I have a particular locus, right? which is often hundreds of thousands of kilobases, right? And you cut it, okay, with some enzymes that cuts every 1,000 bases, you have 100 fragments there. So if I look at a particular locus, like the famous beta globin locus or some other locus like that, I have a 100 by 100 matrix, okay, right? If you want to look at the whole human chromosome, right, and all possible interactions there, so I have 3 billion by 1,000, so 3 million possible such fragments, okay? And 3 million square, I'm not going to try to calculate, is a big number, we all agree, okay? So in theory, there are a very large number of potential interactions that can be fit into the data. The most naive maximum entropy guy would try to fit 3 million square pieces of data where many of these boxes are occupied by almost nothing. Okay, occupied by nothing is maybe good. Actually, maybe not good, okay? Right, I'll come to that, okay? So I again have a variable selection problem at hand here, right? And that has not always been, so I guess there are some people in the maximum entropy community who actually ask the question, okay, of what to do about variable selection there. Others say, you know what, you will get some answers, threshold it, and be happy, okay, right? Also, yeah. I know some numbers. Now, not all of these numbers are very trustworthy. Maybe the big humps are trustworthy, okay? But I don't have a good way. I can go biggest hump, the biggest hump, that will be like in LOD doing what's the best thing, right? But is that the best way? Okay, no, sorry, I lost you.
So you don't trust these numbers themselves that are there?
Nobody should trust any numbers in biology fully, okay? You should trust it only so much.
So what you're saying is you want to know which of them are real contacts and which of them are not real contacts.
Yes, yes. Okay. For example, it's very hard to, you can see the quality of, this is not sequencing data. I think this is, and actually this is partly post-PCR, it could even be sequencing. If you see, I'll argue, if you see a hump there, you expect the neighboring sites to have higher contacts too. Okay? Those may not be their own contacts. Okay? I'll come to that. Okay? Right? So definitely you should not try to fit everything with an interaction. Okay? So what are the genuine contacts there? Right? And I hope that is more or less sparse. Okay? Fine. So what happens, what has been done for this kind of problem? Okay? So what happens usually is that some brilliant guy has some brilliant experimental method to get to the answer. Then you are under time pressure to publish in Nature or Science. And you usually call up, either you know something or you call up somebody saying that, do you have anything to analyze this data so that it actually looks something more than just a bunch of numbers we have gotten, or we just call some peaks, okay? So I suspect in the very first, very important work, among people, Job Dagger gets a lot of credit, but there were some other people there, right? What must have happened is that, okay, you know something about contacts, okay? From contacts, I want to say something about the chromatin structure, okay, right? What do you remember elsewhere in molecular biophysics where from contacts you go to structure, okay? Well, protein NMR, right? In protein NMR, you say you can actually look at shifts caused by a crosstalk between some nuclei, and you know which distance they are, and you kind of put that distance as a constraint and come up with some kind of protein structure. Some cases you can do that, okay, right? So why not take that, right? So someone did precisely that kind of thing. They tried to make a model saying, I'll take one polymer configuration where the distance would be related to contact strength in a certain way, and then chromosome 3 of yeast would look like that, okay, based on the data. And, well, that's a nice picture to have on your web page, plus in the paper, plus maybe on the cover of the magazine, okay? And I'm done, okay? Now, it's not that people who did that don't know that, of course, that chromosome, even if it is looped, it's dancing around, okay? So what you might need is a better description, okay? So then there are many computer scientists who are actually busy saying, how can I make efficient the algorithm to find such structures, okay? But anyway, if we say that this is essentially probabilistic, right, I need a nice probabilistic distribution and hopefully a sparse probabilistic distribution. Not the probability distribution should be sparse. It's just that there should be few important parameters in the probability distribution and no more, okay? So this problem looked like at something to us, okay, which is ready for a maximum entropy type description. Why? Because what you are being told is that the polymer has some kind of configuration space. It has a fluctuating thing, so there is a probability distribution. You are being told how often two things are in contact, so put a fat-end delta function, okay? Its average is what you are being told, maybe up to some biases, okay? So if I give you the expectation of the contact indicator, what can you say about the probability distribution, okay, right? So what should I do? Okay, maybe I think of this chromosome as a bunch of ball, right, one to the other. And I say, now you told me about contacts, I'm going to do maximum entropy, okay? If I do the naive maximum entropy on this problem, okay, what kind of model would I get? So I broke up the chromosome in a few bunch of points, okay? And I said point A to point B contact strength is this much, point B to point C contact strength is this much, right? And I solve my maximum entropy. Well, what you'll end up finding is that actually you are getting a model which has some enhancement of probability when two things are in contact, but that's actually a model of a gas of balls, okay, where potential of the gas has been determined by the contact strength between IJ ball interaction. So I have lost the polymer, okay? So I need to sort of reconstruct back the polymer, okay? And well, if you think about it, you can do that, right? But actually I've hopped on. Maybe I'll just come back to this later, okay? So you ought to modify your maximum entropy principle there by saying that I want to minimize something like a Kullback-Leibler distance from a free polymer to this one. So I want to put in my bias that this is a polymer, okay? If you do that, now you get an interacting polymer. You are happy, your parameters to fit. Remember, entropy is the related to Kullback-Leibler distance from the uniform distribution, okay? So uniform distribution of these points would have been like free gas, and you ended up getting interacting gas as a result of doing maximum entropy. If you put in a polymer, you'll end up getting an interacting polymer, right? But the key thing is that I have too many of these Vij's, okay? I have this n square Vij's to determine, and then what should I do? Okay, and to come back to Sanjay's point, maybe something I was trying to explain, okay? This is sort of the key intuition, okay? I have chromosome positions versus chromosome positions. Well, actually, it's done kind of like that picture. So chromosome positions here and chromosome positions there. Near the diagonal, I always get some bump. Close by fragments end up interacting more, right? That's not a surprise. It's most probably not biologically regulatory anything, okay? Close by fragments of polymer should interact with each other, even non-specifically, okay? So that they get stuck with each other is a good sign that actually at least physics is being followed, okay? The ones I want to spot, okay, are a little hump somewhere else, okay? That's caused sometimes by a contact like that, okay? Now, realize that when these two are in contact, points close like C and D, which are close to A and B, okay, are often in contact with each other just non-specifically. So if I pinch somewhere, close by things would often end up touching each other, right? So it's not surprising that I will see elevated signal from many of the neighboring boxes, right? And it's my job, at least in a sparse model, to ask, can I actually give only one parameter for this whole hump, rather than trying to fit a parameter separately for each of these boxes, which is the naive method, right? Then I would just, and roughly speaking, seeing if I use the same polymer model for everything, I will fit the close by looping probability from the diagonal region, and somehow then given AB has pinched, the CD contact probability will be fit by that shortened distance profile. Okay? So that's the rough idea, that if I fit this whole thing together, I'm learning something about distance-dependent non-specific contact from near the diagonal values, and applying that to this hump to explain everything else. I say introducing one variable explains almost everything. I don't have to separately learn every other thing, and use the polymer structure. Okay, so we, okay, okay, so we, yeah, so the other thing I want to say is that just if you look at dark splotches there, right, you often cannot tell the difference between many different possibilities. For example, if there are 3 sites like this, the red, yellow, green, suppose they're all clumped together, always stuck together, right? You will see a good 1-2 spot, a 2-3 spot, and a 1-3 spot, right? And somehow in here, I could actually have them meeting pairwise but never together. And as far as the patterns of maxima on that graph, they will be the same, right? I can't distinguish the two, right? But if I could compare in this kind of made up example, the relative weights of this whole peak compared to what happens to close by things, I should be able to say something about the strength of the interaction. Okay? Of course, this is very noisy data, and we may actually not have too much of luck. But, okay, that's the hope. Okay, so this is something technical. Rather than having to do full Monte Carlo, one can use some polymerish things to simplify the calculations. Okay, again, one of these slides I'll jump over. This is what we begin to get for East chromosome 3. The real data, okay, looks like something like this. Okay, you start with some model that explains some of these interactions. Then you introduce some interactions, you sort of do it step by step, and you discover some contacts that has some high probabilities of being in contact with each other. And then you can ask in a single cell observation, can you sort of see at least roughly similar probabilities? Okay, so that's all very good. This is where the project started getting into troubles. You have many possible interactions, even in East genome, and forget about human genome, right? And at some point, we tried initially to do exactly the thing that comes to you first, greedy method, okay? Look at where the biggest discrepancy is first, introduce an interaction there, then try to do something, and then you kind of realize that in some cases we actually know interactions we are missing because a local bump has been given too much of importance. Okay, so then we started saying, okay, we'll do something like forward method. We add a parameter, we let go a parameter, I go back and forth, and it got rather confusing at some point. Okay, sometimes it will go actually in circles once you start doing this. So here, so much as by hand, if I say run it up to k interactions, I get some answer that looks biologically interesting. We actually need somewhat more principled way of introducing one after the other. And I'll end by saying that you are most probably guessing where we go, okay, based on what I have said there. So you actually can take an alternative formulation. This is not exactly maximum entropy, okay, but it has exactly the same result. You take log of the partition function, which depends on, okay, so I think I have not explained that. There's a function that depends on all these parameters. You stick in your data this way. Turns out if I vary with respect to Vij, the equation I get, if this term did not exist, okay, is that the expectation of, okay, contact should be equal to data, right? But then I put a penalty, right, for introducing superfluous ones. And then there are many methods of solving this problem. Turns out that actually this is a convex function again, right? Only trouble is that since once I have many contacts, estimating that convex function is a bit problematic, but there are kind of ways around it, okay, at least in practice. So you can now do something like what's called a homotopy method, right? You start with very large lambda. If you start with very large lambda, I have asked you before, very big lambda, what's the solution? What kind of V's would you get? Then I just minimize this function, right? All V's are 0, okay? As I lower lambda, one by one, okay, good V's start coming in. Okay? And you can do exactly the kind of thing I was saying you can do for large or stage-wise regression. You can do it in small steps. You can let in one V up to sometime certain V works, then you say, aha, one more works, right? And this is why it's called like homotopy method. You slowly move through the space of parameters to arrive at wherever you want to stop, okay? And you see that at some point, okay, you have to go down quite a bit before new variables come in where you are beginning to fit kind of noise, right? So anyway, so we actually have gotten reasonable preliminary things in simulated data. In real data, there are other normalization problems to solve, but I'm hoping that this one actually finally gets us somewhere. And therefore, I think I should finish, okay, by giving an overall summary of these lectures. I think I talked to you about importance of sparseness in some space or the other in biological modeling. Okay, there is sort of, I tried to show there is interesting overlap between some ideas from statistical physics and data processing or data analysis here. We are beginning to understand limits of some of these sparse reconstruction algorithms. And hopefully, I have convinced you that there are many potential applications in biology. And thank you. I'll take some questions here. Yeah. Mm-hmm. Mm-hmm.
So ideally, what you want to do is to regularize it that if you have a non-zero parameter, then the close by parameters are zero, right?
Right.
Necessarily achieved by N1 or
But it almost gets forced on you anyway, because once introducing one interaction, so you are saying, should I actually have a correlation hole, right? If you have a parameter there, please don't put any parameter nearby. The point is that if the nearby interaction with the polymer physics around it explains the rest of the bump, the cost of introducing a new variable there is probability enough not so that you actually do the next one very far away. Yeah. There are actually real cases where many things are very clustered. So in those cases, you hope that over the data, over the noise in the data, you can see it. Yeah. Yeah. Okay. It's been a long